Understanding autism. Autism, or autism spectrum disorder, ASD, is a neurodevelopmentally condition that affects how a person communicates, interacts socially, and experiences the world. Brain differences. People with autism process information differently, often due to variations in brain connectivity. Communication and social interaction. Challenges may include difficulty with verbal and nonverbal communication, understanding social cues, or forming relationships. Repetitive behaviors, such as routines, rituals, or repetitive movements, sensory sensitivities. This is heightened or reduced responses to sounds, lights, textures, or other sensory input. Spectrum nature. Autism is called a spectrum because it varies widely. Some may need significant support, while others live independently. Strengths. Many people with autism show exceptional focus, creativity, problem-solving, or unique talents. Causes of autism. Some of the common causes of autism are as follows. Environmental pollutants. Autism spectrum disorder, or ASD, is a complex condition with many contributing factors. While genetics play a role, environmental exposures, especially during pregnancy and early childhood, are increasingly recognized as important influences. Let's look at some of the key environmental contributors. Air pollution. Studies show that children whose mothers were exposed to high levels of traffic-related air pollution during pregnancy are at a higher risk of developing autism. Pollutants like nitrogen dioxide and particulate matter can interfere with brain development. Food and heavy metals. Exposure to aluminium, mercury, and lead, often found in contaminated food, water, or packaging materials, has been linked to neurodevelopmental disorders. These metals can cross the placenta and affect the developing brain. Pesticides. Prenatal exposure to agricultural pesticides has been associated with increased autism risk. These chemicals may disrupt hormonal balance and interfere with neural signaling during critical stages of brain development. Vaccines ingredients and thimerosal. Thimerosal, a mercury-based preservative used in some vaccines, raised concerns. Extensive research has shown that there is a direct link between vaccines and autism. Thimerosal has been used in most childhood vaccines as a preservative. Recent research has identified several environmental pollutants that may increase the risk of autism spectrum disorder, especially when exposure occurs during pregnancy or early childhood. These pollutants can interfere with brain development, trigger inflammation, and cause genetic or epigenetic changes. Pollutants linked to autism spectrum disorder are as follows. 1. Air pollutants, nitrogen dioxide, and carbon monoxide are linked to increased ASD risk when mothers are exposed during pregnancy. Particulate matter like PM2.5 are associated with brain inflammation and oxidative stress. PM2.5 are the smaller size dust or organic particles present in the air, especially during the haze. Heavy metals. Lead, mercury, and copper are known to disrupt neurodevelopment and are significantly associated with ASD in multiple studies. Industrial chemicals, polychlorinated biphenyls or PCBs, are strongly linked to ASD. Phthalates, which are softeners for plastics, are found in plastics and personal care products. Traffic-related pollution. Living near highways or areas with high vehicle emissions during pregnancy increases the risk of ASD in children. How these pollutants affect the brain. Now let's explore how environmental pollutants actually affect the brain, especially during its most vulnerable stages, the fetal development and early childhood stage. These pollutants don't just float around harmlessly. Once inside the body, they can trigger a cascade of biological disruptions. It causes oxidative stress. Pollutants create harmful molecules called free radicals that damage brain cells. Children with autism are often found to have higher oxidative stress, which can worsen symptoms like anxiety, hyperactivity, and poor focus. It triggers neuroinflammation. Pollutants overstimulate the brain's immune system, causing chronic inflammation. This ongoing silent fire can affect brain circuits linked to social skills, emotional control, and language development. Lead to epigenetic changes and mutations during fetal development. Pollutants can alter how genes are switched on or off in the developing fetus. These changes don't always alter the DNA sequence but affect how the brain develops and functions. In some cases, pollutants may even cause mutations that increase the risk of autism spectrum disorders. Heavy metals, lead, mercury, and copper, known to disrupt neurodevelopment and are significantly associated with ASD in multiple studies. Mechanisms of mercury toxicity. 1. Oxidative stress. Mercury binds to sulfhydryl (SH) groups in proteins, disrupting antioxidant enzymes like glutathione peroxidase, superoxide dismutase (SOD), and catalase. This leads to excess reactive oxygen species or ROS, damaging DNA, proteins, and cell membranes. Result: cell death, inflammation, and organ dysfunction. Mitochondrial damage. Mercury impairs mitochondrial enzymes, reducing ATP production and increasing membrane permeability. This disrupts calcium ion homeostasis, leading to neuronal dysfunction and apoptosis or programmed cell death. Neurotoxicity. Methylmercury crosses the blood-brain barrier and accumulates in the brain. It disrupts the following. 1. Sodium-potassium ATPase activity, causing membrane depolarization and calcium influx. 2. Muscarinic cholinergic systems, affecting neurotransmission. 3. Glutamate balance, leading to excitotoxicity. These effects result in cognitive impairment, motor dysfunction, and sensory loss. 4. Epigenetic and genetic damage. Mercury can cause DNA mutations, chromosomal aberrations, and polyploidy. It interferes with gene regulation, potentially contributing to developmental disorders and cancer. 5. Immune system disruption. Mercury affects T cells, B cells, and macrophages, triggering autoimmune responses. It promotes inflammation via ROS and cytokine release. 6. Developmental toxicity. Mercury exposure during pregnancy can impair fetal brain development, leading to cognitive delays, motor dysfunction, behavioral abnormalities. This is especially true for methylmercury, which is efficiently absorbed and crosses the placenta. DNA mutations, chromosomal aberrations, and polyploidy. It interferes with gene regulation, potentially contributing to developmental disorders and cancer. Symptoms of mercury poisoning are quite similar to symptoms of autism, even though they were claimed to be not related.